Okay, good morning. Let's see if I can turn the volume up here. So
The surface of the cell membrane is very, very busy.
Why is this? Wait a minute. What is going on? Okay, can everybody hear me this morning? Everything working okay? We've had some issues all week long with Zoom, so I'm hoping everything goes okay today, but you never know. Anybody have any questions on anything? I've been trying, I've received a lot of emails. I've been working my way through those to get answers out to you on your questions. You know, just be patient. I'll get all them done by today, just in time for more questions. That's okay. Yeah, so any questions I can answer right now?
When did you say the lab was open to like look at bones? Is it every day or is it certain times?
It should be every day. I don't think there are any classes in there. I will go over to campus today to check on that to make sure that's going to work. Just because I set it up a couple weeks ago doesn't mean it's going to be working now. But I will go over today. But nobody should be scheduled to be in that classroom during the day.
Okay.
So, but I'll double check that for you today and let you know. Okay.
All right. Thank you.
You're welcome. Any other questions? Okay. Now, if I remember correctly, we finished up chapter 2. The chemistry finally finished that on Monday, and we did not start chapter 3 on Monday. That's correct. I think that's, at least I checked the video. We didn't go past the end of chapter 2. So I'm going to start chapter 3 on cells today, and we'll go from there. Hopefully I'll do everything right as far as connecting. Let's go into, so everybody should see this. Wait, what? Yeah, there we go. You should all see the slide that says chapter 3, cellular level of organization. And let me minimize that and go from here. So hopefully you can hear me, and hopefully you can see the slide that says the cellular level of organization.
Yeah, I can see it.
Great, that's great. So it's been a challenging week with Zoom, and it's only Wednesday. So okay, and a couple of points about cells. We're going to take a close look at the human cells. It wasn't until the 1850s that it was discovered that everything was made of cells. The whole concept of a cell wasn't really well understood up until then. Actually, the term cell goes way back. It goes back to like the 1600s, when early anatomists were using their primitive microscopes to look at plant cells. They were looking at slides of cork. And one of them said, wow, that looks just like jail cells or cells in a monastery, little rooms the monks would live in, one after another. And so the term cell came from that, believe it or not. That's really why we call them cells now. And I'm sure that's gonna, I've never put that on the test, by the way. So the 3 key points here: all living organisms are made up of cells. from the simplest one-cell amoeba to a human, we're made of cells. Bacteria are made of cells. Funguses are made of cells. Protozoans are usually just a cell. Bacteria can be many cells linked together, but they're all living cells, and only living. And viruses are not a cell. I'll just emphasize this: a virus is not a living organism either. A virus is nothing more than a coat of protein that surrounds a simple strand of DNA or RNA. All a virus is programmed to do is to inject its DNA or RNA into the nucleus of a living cell, and then take it over and direct it to make copies of the virus. And that's all a virus does. So everything living is made up of cells. A cell is the smallest unit of life that you can have. There's nothing smaller than a cell that's going to be alive, whether it's a bacteria or a fungus or a protozoan. If it's a cell, it's alive. If it's less than a cell, it's not alive. That's why a virus isn't alive. So all living things are made of cells. Cells are the basic unit of life, nothing smaller than a cell. And the most important point here is that living cells only come from other living cells. You can't artificially create a living cell. There's no such thing as insta-life or proto-life that you can mix together with, some carbon and hydrogen and oxygen and some nitrogen and get living cells. So they only come from other living cells. Now, in our bodies, we have well over 200 different types of cells. They're all specialized to do certain things. They have different shapes.
They can't see you or nothing.
You can't see?
Mm-mm.
You can't see the slide.
No, I just didn't want to wake him up.
Hello?
Yeah, I'm not gonna do that.
Okay.
I can see it on my part.
Okay, that's good.
I was gonna.
So let's put, okay. Now, in our bodies, we have over 200 types of cells. They all have different shapes. They can be a sac of hemoglobin that we call a red blood cell, because that's all a red blood cell does is carry hemoglobin to carry oxygen. They can be specialized cells like the neurons, and some of these neurons can be 3 or 4 feet in length. The nerve cells can be 3 or 4 feet in length, because if you have a long nerve like the sciatic nerve that leaves our spinal cord at the hip and goes all the way down to our big toe, that's made of thousands of neurons linked together. Some of the cells are free-floating. Most are bound to each other. Some are column-shaped, some are circular, some are cuboidal, some are squishy. Like white blood cells are squishy. They act like an amoeba. They all do different things. They can be layered, or they can be multi-layered, or they can be a single layer. So we have this wide variety of cells going on here. Now, regardless of its cell type that we see, they're all going to have 3 general structures that are the same. Now, the erythrocytes, red blood cells, are the exception to this, because the 3 general structures are a plasma membrane, cytoplasm, and a nucleus. Red cells don't have a nucleus. They did when they were developing, but they kicked their nucleus out to have more room for hemoglobin. But every cell is going to have a membrane. It's going to be the plasma membrane. It's the edge of the cell. When we've looked at slides of cells, even those slides of the cheek cells I showed you, it had a membrane. You can see where the membrane's located. You couldn't see the membrane. It's too small for that, but you could see where it was located. The cytoplasm is the cell goop or cell sap inside the membrane. And if you look at like cheek cells stained blue, well, that's the cytoplasm. Or some books now are saying cytosol. You know, the names change over time. 70 or 80 years ago, an old textbook would refer to it as protoplasm. Cytoplasm became the word to use in the 1970s, 1980s, somewhere in there. And now we're gradually seeing a transition into the word cytosol. You know, it all means the stuff, the sap, the material, the fluid portion inside the membrane. And then other than the red cells, every cell has a nucleus. That's where the DNA is located in our cells. Now, the DNA inside a nucleus, if every cell is identical, they all have the same information. All of our genetic information is contained on that DNA. But if you're a cheek cell and you're compared to a nerve cell, well, the information on how to be a nerve cell is turned on and everything else is turned off. If you're a cheek cell, how to be a cheek cell is turned on and everything else is turned off, but it's all there. All the information is there. Nothing is left out. It's just not being used for that particular purpose. So your sort of a typical type cell looks something like this. Not everything's going to be present in every cell, and they're all going to have different shapes. But we see a membrane, that's this layer right here, that is going to be selectively permeable.
No, I'm okay. Thank you, though.
It's going to be controlling what comes into the cell and what goes out of the cell. It's very, you know, we say it's selective. It will dictate what can come in. And what comes across the membrane is very, very important to a cell. Cells are very, very particular about what they let in, and even more particular about what gets out. And so we see the membrane, we see the big purple thing here is the nucleus, and then we have, these are all organelles inside the cytoplasm. And many of our cells are going to have these organelles. Some are going to have more than others, depending on what they're going to be doing. But this is sort of a generalized structure. Now, many cells are going to have these folds on the ends here. Our simple columnar cells that we find in the small intestine are going to have these microvilli to increase their surface area. We want to be able to absorb nutrients in the small intestine. So we increase our surface area by these little folds of tissue, folds of cell membrane here, so we can increase the surface area tremendously. We don't have to see these kind of folds on a nerve cell. We don't see these folds on a muscle cell. We only generally see them on those simple columnar cells. The membrane is made up of phospholipids. Same phospholipids we talked about in chapter 2. It's a triglyceride. You know, it's a 3-carbon alcohol with 3 fatty acid chains attached to it, but the 3rd fatty acid chain is removed and a phosphate is placed on there. And this forms what we call the lipid bilayer. The membrane layer of the cell is a double layer with phosphate heads on the outside and on the inside. The fluid bilayer will suspend numerous proteins embedded in the layer. These proteins are going to float through here. They serve as passageways for the movement of nutrients and water and ions and other molecules. They identify these proteins that are on the surface of our cells, identify us as self. These proteins, when they're combined with a carbohydrate, form something known as a glycocalyx, and that tells the cell that, hey, I'm me. You know, I'm one of yours. It's an identifier. You know, it tells you the glycocalyx. Every cell has glycocalyx structures that are recognized by the body's immune system. So we know that every cell belongs there. It's not foreign. When a patient has an autoimmune disorder, like lupus, for example, it's because the immune system fails to recognize those glycocalyx structures and attacks that particular organ. And we also have ways to hold the cells together, because the cells don't just randomly float free inside our bodies. Many are grouped together like muscle cells and stratified epithelial layers, like in our mouth or in our rectum. So, typical membrane, the animation isn't that great, but this is what the our phospholipid bilayer looks like. We have the phosphate heads here, and we have the phosphate heads here. And since we've eliminated one of the fatty acids, there's only two here. The fatty acid tails are in the middle. But this gives us the equivalent. This gives us a double layer of these phospholipids.
The plasma membrane is selectively permeable. It lets some substances through.
The middle of our cell membrane is water hating. Nothing gets through that's dissolved in water. Now, things that are dissolved in lipids, in fats, can slide right through. Cholesterol, a steroid, it slides right through the membrane. In fact, the cell uses steroid for reinforcing structures in its membrane. But steroid-based hormones can slide right through here. Anything that is soluble in a lipid can go through the membrane. The membrane is lipid loving. Vitamins that are fat soluble vitamins, like vitamin E, for example, or vitamin K, or vitamin A. These are all going to be fat soluble, and they're going to slide through the membrane very, very easily. So it's things that are dissolved in water, like vitamin C. Vitamin C is water soluble. It can't go through. It needs help to get in. Glucose, sugars, can't get through the membrane. First of all, they're big molecules, and second of all, they only dissolve in water, and they are not, anything that's water soluble has a real hard time going through the membrane. So if we want to get sugar into our cells, we have to open up special channels just for for it to get through. Oxygen, not a problem. Oxygen molecules are so tiny they just slide right through. The same with carbon dioxide; they slide through going out. But sodium is a small structure, but it's an ion. It has a charge, and that charge is what's holding it up. Any of these ions aren't, because ions are all water soluble, ions are going to have a hard time getting through the membrane. So we have to have specialized channels for them too. So the membrane is very, very selective as what's going to go through here. Okay. So there's your phospholipid structure. That's what it looks like. The big round thing is the head. That's the phosphate group, but it still has the 3-carbon glycerol. And we have 2 of the fatty acids down here. Now, the fatty acid don't join together like we do for a solid fat, because there's one of the fatty acid chains is unsaturated. So we have, it kinks and bends and twists and rotates at the double bond. So it maintains its liquid state. But this is this is what the phospholipids look like. And again, it arrays itself so that the tails, the fatty acid chains, are in the middle of the membrane, of the cell membrane, because the membrane is a double layer. So you have the phosphates on the inside of the this layer facing the inside of the cell, and these structures here facing the outside of the cell. And the entire membrane looks like this. The entire membrane is a very squishy surface. It's like trying to walk on a trampoline surface. You know, if you ever go to one of these trampoline facilities in Sevierville, you know what trying to walk on that surface is like. The surface of the cell membrane is like a water balloon. It's very squishy. It rebounds, and you press on one side, another end pops up, and the proteins that are embedded in the membrane, like this is a protein. This is an integral protein. This is called a peripheral protein. Proteins embedded in the membrane are going to slide around through here, as will these proteins with carbohydrates attached. This is a glycoprotein. Glyco for glycogen or glucose, and then the protein. This may be a glycocalyx. Over here is a carbohydrate with a fat attached to it. Sort of the funnel cake of the cell. It's the sugar-coated fat, if you will. I'm not picking on funnel cakes. We have cholesterol embedded in the membrane for reinforcement to give it strength. The whole thing is going to move up and down. It's going to be, again, like a water balloon. It's very, very, very squishy surface. You know, and imagine a water balloon covered in olive oil, rolled in a cereal, and the cereal can float on the surface of the water balloon. It's going to move around. So the cereal is suspended in the oil, and it's going to float on the surface. And that's what we call this the fluid mosaic model, because it's always moving around. You know, we will open up these proteins as we need them. The proteins are what we call channel. Sometimes they're channel proteins. Sometimes they're carrier proteins. They're there to facilitate movement through the membrane. Other proteins are there as enzymes or as reactive sites. So they're constantly on the move. But most of the membrane, 75%, are the phospholipid structures. Now, a good portion of that membrane is also cholesterol. So a person that would say, again, you know, back to that comment I made earlier, a person that would say that I'm never going to eat cholesterol again, I want to keep it totally out of my diet because it's not good for me, you know, so I will never eat it again, is doing themselves a disservice, because our bodies need a certain amount of cholesterol. We need it to make the steroid-based hormones. We need it for the reinforcing cholesterol in our cell membranes. So we have to have cholesterol in our bodies. So it is not a function of, you know, if some is bad, none is better. That's not the case. So, okay. So now the proteins in the membrane are pretty active. The proteins are used to communicate to other cells. The proteins are used to hold cells together. They serve as reactive sites, and they serve as passageways for the substances that the cell needs, but the membrane won't let it through, like glucose, sugar. The cell needs glucose. That's its source of fuel. Our cells depend on glucose so that we can get energy. Glucose is a big molecule. It's also water soluble, and it can't get through the fat layer. However, if we have a specialized, what we call a carrier protein, that will allow glucose and only glucose to come into the cell, then we have a workaround on the membrane. And this is what insulin does. Insulin, you know what insulin is. Insulin is what controls the our blood sugar levels. And when our blood sugar goes up, we release insulin from our pancreas, and that brings our blood sugar back to normal. The way the insulin works is insulin stimulates cells to make more proteins called a carrier protein that will allow sugar to come in. It will allow the sugar to come in, and then it'll stop making the it will stop. The insulin doesn't do it, but it doesn't make the proteins, but it stimulates the cell to make these carrier proteins. And once our blood sugar comes back to where it belongs, to our normal level, then we'll stop releasing insulin and the cells will stop making these carrier proteins. Because it's only a certain amount of sugar our cells can take. Anyway, that's a workaround, and it works just fine for us. Other proteins function to allow water to come through, or sodium to come through, or potassium to come through, or calcium to come in, or allow certain amino acids to come in. So there's all sorts of advantages here to having these specialized proteins. Now, a protein can be an integral protein, meaning it's embedded and goes all the way through the cell membrane, or it can be what we call a peripheral protein, embedded on one side or the other, sort of tied to the integral protein. So what you're seeing here in this little animation is a integral protein that extends all the way through. Usually, the integral proteins are going to work as some type of transport protein. It can be a channel protein or a carrier protein. Some of them function as enzymes. Some function as receptors for hormones. The hormones only land on cells where they have specialized receptors. And so these integral proteins act as a receptor. The peripheral proteins are usually tied to the integral protein. They're only on one side or the other. They work as enzymes. They work as connectors for cell to cell, because the cells are tied to most of the cells are tied to each other, and they function in muscle contraction. Our muscle cells are made up of what we call contractile protein. In skeletal muscle, we'll see when we get into chapter 9 that our muscle cells slide across each other to contract. And it's the ability of these cells to move across each other that causes this, contractile, condition, contractile ability, if you will. So we refer to them as contractile proteins. Now, you've seen one of these in action during cytokinesis, the stage that occurs after mitosis is finished. Starting in anaphase and continuing through telophase, cytokinesis occurs because the actin protein, it's called actin, starts squeezing the cytoplasm in half. So you end up with two distinct daughter cells. So this is, that is a peripheral protein doing that for us. Okay. So there's a variety of things that proteins do for us, transport and enzymes and holding cells together, recognizing cells. The cells combine with a carbohydrate to make the Glycocalyx. And I'll show you some of these here.
Transport proteins carry molecules from place to place.
The hemoglobin is is not the type of protein that you see embedded in the membrane completely, to allow nutrients to pass back and forth. hemoglobin is a very large protein molecule that floats inside our red blood cells carrying oxygen, but it's still a transport protein. Because that's how we move oxygen. Red cells absorb oxygen that binds to their hemoglobin, and the hemoglobin, which is packed inside every red cell to the point where the cell is almost ready to pop. The hemoglobin inside the red cell carries a tremendous amount of oxygen, and that's how we transport the red cell throughout our bodies, and it's in our blood is in contact with every cell, either directly or indirectly, and oxygen will leave the red cell and go into the tissue itself. So over here we see now one thing we see here, and this will make a lot more sense when we get into AP2 and look at hormones. But hormones, if they're protein based, are very large molecules. They also can't get across the membrane. In fact, there is nothing that will allow a protein hormone to cross the membrane of the cell. But what a protein hormone does is it lands on its receptor, which is also a protein, and that receptor becomes stimulated and passes a signal on to another protein embedded in the membrane, which then finally allows a signal to go inside the cell. So we call it a second messenger. The hormone's the first messenger, that's this blue thing here, and the second messenger is this green thing. It's a workaround to get the information from the hormone into the cell, because the hormone itself is just too large to get in. Here we see structure. Many of your structural proteins are going to be the alpha helix or beta pleated sheet structure. Holding the cells together, giving our cells their distinctive shape, an extremely important role of the of proteins. And in this last one, we'll talk. So we know how important enzymes are. We've talked about that already in the chemistry section, but many of these enzymes are can be embedded inside, in or on the cell membrane. Now, Glycocalyx structures. These are the identifiers. They are carbohydrates usually attached to a protein, occasionally attached to a lipid. They are the sugar coating on these structures, but they are what we call a biological marker, and they allow the cell to be identified as self, meaning us. So every cell in our bodies has the same antigens or same types of antigens, all of it being identified. And very early in life, usually within the first year of life, our immune system trains the white cells in our blood, which are the white cells are our infection fighters. It literally trains our white blood cells to recognize all of our glycocalyx structures. We call the glycocalyx structures antigens. And these antigens, while they may all be different on different cells, are all glycocalyx structures, and they are all self-identifiers. A failure to identify self is what is the whole basis of an autoimmune disorder. You know, when we attack our own cells, we're attacking our cells because we don't recognize them anymore. And that does happen. You know, it happens in lupus. It happens in multiple sclerosis. It may even happen in diabetes for some individuals. You know, so there are a variety. It happens in the adrenal glands, where we attack the adrenal glands, or we attack the thyroid gland. And there's all sorts of types of autoimmune disorders that we can encounter simply because we fail to recognize our immune system fails to recognize self, our own cells, and it happens. Now, usually it doesn't happen. Usually, you know, we don't have that kind of issue, but sometimes we do. So, but it is all these glycocalyx structures are also the basis of identifying blood types. We have, in addition to the self-identifiers, we also have blood type cell identifiers that tell us what type blood we have: type A, type B, type AB, type O. And our immune system, our white cells, recognize these identifiers and say, "Oh, well, that's us. You know, that's our type of blood. We can't, you know, if you're type A blood, you can't take type B blood without, you know, some very serious complications, maybe sometimes even fatal. If you're type B blood, you can't take type A. So there are some real concerns here to make sure that we don't give a patient the wrong blood type. Blood typing is very critical. It can have, you know, it can have fatal consequences. Cancer cells can take advantage of our immune system. Cancer cells, well, first of all, cancer is nothing more than uncontrolled cell growth. That's the basic definition of cancer is uncontrolled cell growth. The cells don't keep, normal cell growth is kept in check. Our bodies are designed to not let cells overwhelm one area or another. We normally don't have uncontrolled cell growth, but a cancer cell removes all the safeguards, and it will have uncontrolled and very rapid, usually, cell growth. And it will have foreign glycocalyx structures on the surface of the cell. Now that should trigger the immune system to respond to it, to attack it, to destroy it. But what the cancer cell does is it keeps changing its glycocalyx shape. It changes it to a new one very rapidly. Now our immune system takes about 10 days to 2 weeks to respond to a foreign antigen. It has to recognize it, and it has to start preparing to respond, then it responds. And so it takes a 10-day to 2-week period. Example, if you get the flu, if you were to get the flu, and you would be, I'm getting power fluctuations here. Can you all still hear me?